Okay, we're recording now. Thanks, Katie. All right, you're good to go.
Okay. Sorry, folks. So my project is working with modeling soil organic carbon content stock in the eastern Allegheny Plateau and mountains. Skip had talked about that region in his presentation, but basically it extends through 5 states: New York, Pennsylvania, West Virginia, Virginia, and Maryland. It's a very diverse region. It has a wide range in elevations, in precipitation, mesic and frigid temperature regimes, and 7 of the 12 soil orders. So the purpose of this study is carbon has been modeled throughout the United States by many different ways, and the values range from 2.2 kilograms per meter squared to 32 kilograms per meter squared in the upper meter in this region. That's a huge range, and so I wanted to see if we could model carbon using environmental covariate and lab data. So, soil has 3 times more carbon than in the atmosphere, and it has been noted that there's a need to quantify dynamic soil properties in the realm of soil survey, especially during this time of climate and land use changes. So here's a slide of the different studies that have been done in MLRA 127. You can see there's huge ranges for the upper 1 meter of soil. The NRCS products, they're SSURGO and SSURGO. You can see that they have very low estimates of total soil organic carbon. The soils databases usually do not incorporate organic horizons into their carbon estimates, so that's why those are probably pretty low. The rapid carbon assessment, which was an initiative put out by NRCS in 2010 to look at the carbon distribution across the United States and modeling the carbon samples visible near infrared spectrometry. That was done, and there's a higher estimate of carbon with that product. So here are some of the hypotheses of my research: that SSURGO and SSURGO underestimate true carbon stock, that missing Kellogg Soil Survey Lab, which is the national soil survey lab for the United States, that data, there is a lot of missing variables, especially carbon and bulk density, that we can estimate those and use those in modeling efforts. And then soil organic carbon can be modeled using geographically weighted regression kriging as a function of the harmonized legacy data and the environmental covariates to better reflect carbon stock in MLRA 127. So for the missing data in MLRA 127, 50% were missing bulk density estimates and 13% were missing carbon data. I looked at 250 Pedons that have been sampled and georeferenced within that database for this region. The earliest was sampled in the 1960s, and the soonest was, or the closest to now was in 2012. So in order to use these data, certain steps had to be done. So updating the series correlation and then the horizonations and depths. A lot of these soils in the 60s, 70s were described, the O horizons were described as litter and not incorporated into the soil profile. That had to be adjusted. A carbon equivalent correction regression factor was used to convert wet combustion carbon estimates to dry combustion carbon. Traditionally, back in the early days, wet combustion or the Walkley-Black method was used to estimate carbon. It's been shown to underestimate true carbon stock and overestimate in the lower horizons. So in order to correlate the 2 methodologies and just have similar data, we converted the Walkley-Black to the dry combustion estimates. In MLRA 127, about in 1993, that's when dry combustion was starting to be used. Now it's the standard for NRCS for calculating carbon. And then outliers with any sample data and lab analyses, you're going to have outliers, and those should be excluded from your model. And then for rock fragments, back in the 60s, 70s, 80s, rock fragments by weight were not described mostly, so I had to use the rock fragments by volume. Those were described visually in the field by soil scientists. So those were used to calculate soil organic carbon stock. So stock is a function, carbon stock is calculated based on bulk density, carbon, and rock fragment content. So usually, to populate missing data, in the literature they've used pedotransfer functions, and they predicted, say, bulk density, and then used that to calculate carbon stock. So for carbon, if you're calculating stock, a lot of these times, like for bulk density carbon, they would use carbon to predict it. Well, you can't use carbon to predict the factor that you're going to use carbon in the equation with. So, and usually they make implausible normality assumptions, so that was not used in the study. Instead, we looked at the random forest technique that Travis described earlier, and then also mean and median values by horizon and texture. So like an A silt loam or B silty clay loam, those values of the data. So some of the results. Mean horizon by texture with outliers included actually performed best for soil organic carbon prediction, and random forest did best for bulk density. Organic horizons were the main source of error. Not a lot of data exists on organic horizons in the database, so you were trying to predict a lot of missing data from a very small data set. And there's a lot of variability within organic horizons. Random forest is the preferred method for populating missing data due to non-parametric technique and simplicity. And then future research is needed to detect and account for overfitting in random forest models. So random forest tends to fit noise in the data set, and there currently hasn't been too many methods to get around that. So addressing that in the future is important. So similar to what Travis had showed, the variable importance for bulk density, horizonation, depth, taxonomy, texture, color were the main important variables for predicting bulk density. And for soil organic carbon, color was more important than taxonomy. So digital soil mapping of carbon. There's been several techniques done in the past, usually one of these 6 methods. So multiple linear regression, ordinary kriging, regression trees, regression kriging, geographically weighted regression, and geographically weighted regression kriging. The first 4, according to Kumar and others, 2012, are described as stationary models, meaning that their predictions do not vary over a geographic scale. Of course, carbon will vary based on the location on the landscape, so those were not used in the study. Geographically weighted regression, it accounts for geographic variability, but it doesn't directly consider spatial autocorrelation, which is a fancy term for basically that, data points that are close together should be similar than those that are far away from it. We know in MLRA 127, you have different spots of high elevations. They're not all connected in one spot. So carbon is going to be spatially correlated. So in order to account for that in my model, I looked at the final one, GWRK. It accounts for non-stationary and also spatial autocorrelation. So just to explain kind of how this model works, it's a weighted least squares regression equation, and it looks at the different covariates and assigns different coefficients based on each data point. So each data point has its own regression equation. So go through this equation real fast. The U and V represent the geographic coordinates. Beta is the coefficient for that variable. Alpha is the intercept to the equation, and then X is the dependent variable, and then the error term. And then Y, of course, is the carbon stock, or your variable of prediction. So with GWRK, you can also choose to use a fixed or an adaptive kernel. In this study, I looked at an adaptive kernel where you can see that the kernel size will vary based on the data points and the covariates used in the model. So here's a list of the covariates that I selected to use in the model, but I did not use them all because some were related to others, some were calculated from others. You don't want to incorporate multicollinearity issues in your model. So in order to figure out which ones were correlated to each other and exclude those and see which ones predicted carbon the best, I used random forest, Spearman, Rank, Kendall's, Cow, and Pearson correlation. Those are all statistical measures. So the covariates that I ended up using, the ones in green were positively correlated to soil organic carbon. The brown were negatively correlated. So the lab cut-ons, available water storage calculated through SSURGO, taxonomy, the geocover that Travis had talked about earlier, the DEM variables and derivatives, gamma radiometrics, and geology, climate, and then vegetation indices. So here's kind of a summary of the data set that we had. This is not modeled. This was what actually the Pedon's lab Pedon data. So spotosols that we've been talking about all day have the most carbon, followed by altisols, insectisols, alphasols, and then entisols. Alphasols in this region, MLRA 127 has a lot of poor nutrient poor sites. So alphasols are rich site, and most of those have been cropped. All the ones that were sampled have been heavily cropped. So that's why they have a lower value than you probably would think. But yeah, so that kind of explains the importance of carbon sequestration within spotosols to see that they have the most potential. So here's a result of that map on the carbon prediction map. On your all left, we have geographically weighted regression model, and then this incorporates the kriging. So kriging is an interpolation method, and the difference between, I went over this briefly, but the map on the right incorporates the error term of the first map. That's the only difference between the two. So Skip and I were looking in the office yesterday, and I need to explore these results more, but in this region in Pennsylvania, we were wondering why this was red, why there was a low carbon section here. Turns out these are surface mines that had been done and earlier in the 1900s, and so these soils are entisols. They have different spatial, like the uranium and the geocover. They picked up those soils in this model. And then there's this one speck. It's really hard to see at this scale, but there's a red low carbon spot near Spruce Knob, and Skip was kind of like, I don't understand, Spruce Knob's the highest elevation in the whole MLRA. Why does it have low carbon there? And we looked at it. It's a limestone pasture hayfield. So anyway, we need to further look at this and see what it actually could predict. But it's looking good. But you can see that the R squared is kind of low, and with that big of an area, that's a pretty good correlation. It's a huge geographic area to predict carbon, so with limited data inputs. This is a comparison of SSURGO and StatGO. In SSURGO, you can see the nice county lines, and then StatGO, you can just see these are all at different scales compared to the last slide, but there's a lot less carbon in the R squared. So the R squareds were calculated from a separate validation site. Those sites were from the U.S. Forest Service, and I also analyzed the RACA, the Rapid Carbon Assessment sites, through dry combustion in the lab. Those are my validation sites. So some future work that needs to be done. I need to assess uncertainty in these models. So how certain are we sure that this area has this much carbon and this one doesn't, and all of that. And then determine if the fully populated data set is actually better to incorporate in the model than the unpopulated limited data set. Construct a framework guideline for other MLRAs to do this so that they can understand carbon distribution across their MLRA, and then to further explore the results and applications of this technique in the realm of soil survey. So some applications for USDA to, at least for NRCS, to spatially disaggregate complexes and look at minor components within map units. So currently the SSURGO database and StatsGo2 as well give map units, and they list components. They do not graphically show you where those components are within that map unit. So if you have a wetter component in a drier map unit, you should be able to pick that out based on the carbon. You know, there should be more carbon in that soil. So it helps you give a visual explanation of these minor components. For forest, crop, and pasture productivity assessments, obviously if there's more carbon, the soil, you know, you should be able to assess where the most productive sites are. And of course, like Travis had mentioned, sequestration and land management. So if a place has a lot of carbon, should you really target that area for road construction or for timber harvest? You know, maybe you could go step a few feet or a few meters this way and use that area for construction or so forth. And then NRCS has put out the Soil Health Initiative, which is to educate everyone in the United States and the world basically about soil health, the importance of soil health, why we do the programs that we do. And with this, more, you know, soil health is usually looked at through cropland. You say more organic carbon, that means it's a really healthy soil, and that's true, but each soil has a potential. You know, in these soils that Travis and Skip and Jason were talking about, you're not going to want to plant broccoli and lime it to death. That's just not the health of that. That's not what that soil is designed for. That's not what the historic use of it should be. So anyway, there's a lot to do with that. And then I have several references. And then I kind of want to summarize this seminar today through this picture and quote. The history of the soil is written indebtedly in the soil profile itself. All we need to know is there if we can learn to read it, which I think you've seen is very true. So this soil, or this shot, was taken, these are limestone hills on either side, and this is kind of a broad, flat-ish area for our region. So I don't know, what do you think, what soil might be underneath that? You all? Yes. I kind of gave it away. So this is Blister Swamp, West Virginia, and if I had seen this picture, not knowing where it was taken, I probably would say there would be some kind of a moderately well-drained soil or an alta-salt, something. I would not think that this is the soil that's underneath that. You know, Jason and I dug several plots here, and what Travis had touched on, you have the bisequal. This soil, some of these soils were trisequal. I mean, they had so many feet of sphatic materials. I just never would have thought that, given the cows have been grazing on it for 150 years. So that signature is in that soil. We just, you know that obviously this place was covered in some kind of conifer, balsam fir, perhaps, or spruce, hemlock vegetation at one point. It's definitely not native pasture. So I think this kind of summarized the importance of looking at soils and understanding that the above ground information, the history is below. So with that, I will take any questions.
I have a question.
Uh-huh.
How much work would it take to take the soil organic carbon layer, whenever you finally get there, and to try to actually marry it to ecological provinces or land type associations and so forth, and actually start looking at
Management.
Yes, soil organic carbon numbers related to above ground ecosystem types, and start thinking in those terms, because the Forest Service has a mandate that we will look at carbon as we do planning and as we conduct our NEPA. And I really think that is maybe the approach that we could utilize, that if we understood what our soil organic carbon numbers were, that we know what our above ground biomass carbon numbers are, and then we can marry them to an ecological type hierarchy from a vegetation classification where we have silviculture prescriptions and wildlife objectives.
Yep.
And how far away are we from being able to do that?
I feel like we have a lot of resources that, and with imagery and such, you can kind of estimate carbon spatially. So above ground biomass, the hardest part is below ground. But using lab data that's been sampled, I mean, these estimates didn't come from, someone going out and saying, oh, I think this is it. It's lab sample, it was data that was sampled. And so I'd say we're pretty close, at least to give you a guideline of where you should manage for certain species, perhaps. So the carbon stock that was predicted is not, it's what's there now. It's not what the potential is. It's not, it's whatever is at that site when it was sampled. So between 1960 and now. So. Mm-hmm.
Just a quick question on the difference between your modeling and the stats that SSURGO. I mean, you've got a slide that sort of looking at that difference. I was trying to assess in my mind. Okay.
I'm sorry.
What's the magnitude of that difference? I'm not sure I heard.
Okay.
Absolute amount or percentage difference.
The question was, what is the magnitude and the difference between StatsGo and SSURGO soil organic carbon estimates? Is that
Yes, that's
the
correct.
difference between those two and your model?
Okay. By looking at the means, that kind of gives us a perspective. Here it's about 11.78 kilograms per meter squared, and if you look at StatsGo, SSURGO, 2 and 4, these two are comparable. The red is 0 to 2, 2 to 4, 4 to 6, 6 to 8, and so forth. With the other slide, it was 0 to 5, 5 to 10, 10 to 20, 20 to 30, 30 to 40. So the other one ranged from 0 to 10. This ranged from 0 to 40.
Just looking at the means, it looks like at least factor 4.
Yeah, which, But the rapid carbon assessment kind of did that. It predicted similar to the values that I had. But just looking at the spatial extent of those, there's a lot more variability, little pixel by pixel differences explained by this model that's not in the carbon model. But that needs to be further explored. Stephanie?
So another question I had was, I didn't see any pistosols in the
Right.
presentation. So maybe you can tell us why that is, and maybe what, if you had included pistosols, what would that do to the data set?
So the model wasn't trained with any histosols. Steph was asking, for those on the conference call, if, what about histosols? We do have them in MLRA 127. We have a peat bog really close to where we're at right now, at Canadian Valley, but the model was never trained with it, so it does not necessarily know how to pick out based on taxonomy, but there are other variables such as wetness and geocover that would help pick that out. But if they were sampled, of course, any data, additional data added to this model is going to enhance it because it's going to have a better understanding of where those relationships are. So
It probably blow up the numbers even further for carbon,
probably, because it can understand, yeah.
even though they're small in extent, they're massive in storage.
Storage, yes. That'll be a good thing is to look at these areas, look at where those areas are and see what it did predict and what maybe, if you did sample it, what it would do.
in this process that you use, can you see this being used for population component level aggregated data, component level data?
So currently, in the gridded SSURGO product, Sharon Waltman and others have calculated carbon stock of different depths, and that's available with the current version. But you could definitely do it at a component level, similar to what they had done. So.
Any questions? Okay.
It was 30 meters.
Yeah, resolution.
What was the resolution of the model?
30.
Yeah.
Okay, Sean, you guys, do you have any webinar questions?
There are no online questions.
All right. Well, that, for the webinar, what we're going to go back, I believe anyway, some of the missing slides in Travis's presentation. We had some technical difficulties here. So we're going to go back and review some of the earlier slides, with the group here. So I think that concludes our webinar for today. And I want to thank everybody for attending. And these recordings of these sessions are available. Sean had provided that information at the beginning. Sean, do you want to just reiterate that in case later people that join the webinar?
Sure. And all of these have been recorded, and we go through a file conversion process, make them ready for YouTube, and they'll be publicly available on our NRCS National Soil Survey Center YouTube channel in the next few days. And it takes us a while to process those files and get them posted on YouTube. So you'll be able to go to YouTube, do a search, and it should come up for you.
And if there's any further questions or further feedback or inquiries from the webinar attendees, please contact me, Stephanie Conley, at sconley@fs.fed.us, and I can provide you with your certification, certificates, follow up to anything of the seminars, or contact information for any of the presenters. Thank you.
Thanks, Stephanie. And thanks again, Sean. And hope everybody enjoyed the sessions.
Wow.
Sorry about that.
Yeah, thank everybody for doing this. We know that there's pros and cons to having a live webinar with a simultaneous lecture series, and we opted to do that because I had so much interest from out there that people said they could not make it. Elkins tends to be a remote place compared to not exactly Texas. So I appreciate everyone that came today for your patience with all of our technicalities. Okay, so with that, let's try to go back.
Yeah.
See what
Okay.
Travis